it's time to get started. And we're going to get started with our Masters division. Our first competitor will be out here in just a moment. Our first competitor from the Department of Anthropology is Alyssa Carpenter.
If I asked you what countries fought in the American Revolutionary War, you'd say the British and the American colonists. But what about the French? One of the ways we're learning just how important other nations were for the war is through archaeology, specifically the archaeology of cannons. My research is focused on the conservation and historical analysis of 20 Revolutionary War cannons recovered in Savannah. In 2021, when deepening the Savannah River, the U.S. Army Corps of Engineers unexpectedly encountered a unique assemblage of artifacts. 2 years later, when they arrived at the Conservation Research Lab, they looked like bright orange Cheetos. They were covered in concretions and red Georgia mud. It was hard to identify any features that clearly screamed, "This is a cannon." We hammered and chiseled and scraped all the concretions off of the surface and found that most of the cannons remained loaded, containing gunpowder bags, cannonballs, rope, and wood. The rope recovered looked as if you went to a craft store and bought a ball of yarn yesterday. It's extremely rare for both paper and rope to survive for hundreds of years and from an underwater environment. The bore contents of these cannons provides a unique opportunity to fill in the missing pieces in the historical record. The gunpowder bags were secured with a folded bottom. This is a feature that has never been documented before. My research reveals that there are multiple types of cannon present, but I was still faced with the question of where do these cannons come from and what that meant for our understanding of the war. To assist in the identification, I laser scanned and 3D modeled each of the cannons. I also conducted elemental analysis of the cast iron, which reveals the exact elements present and in return details about their manufacturing process. These guns were ready to be fired within seconds of anticipated battle. The wooden plug could be removed and instant fire could occur. In other words, the Navy ships that these guns were on were ready to fight at any moment. These guns give us a glimpse into the everyday life of the soldiers and sailors on the front lines. The colonists with their young and loosely organized Navy would not have the same effect if it weren't for the additional tremendous aid from other nations. In 1779, during the Siege of Savannah, the presence of both British and French navies were stationed in the river. This collection of guns reflects that. 3 are mid-18th century British guns, and the remaining are what I stated in my research to be either colonial made with French influence or French made. My research has shown how these 20 mysterious, rusted, beat-up cannons from Savannah reshape our understanding of our country's origin and just how global it truly was. Thank you.
So it's time for the second of our 3 finalists in the Masters category, also from the Department of Anthropology. Welcome, Kimberly Breyfogle. Kimberly.
What do you think of when someone reminds you of your grade school history class? For most people, these are not inspiring memories, maybe a teacher droning on about some old war. Now think about when you see or even hold a historical artifact. They make history feel so much more real and give us a solid connection to the people of the past. Archaeologists dig up artifacts to get more information and that physical connection to the diverse people of the past. But usually, artifacts that have been dug up are falling apart, and they need some sort of conservation so that future generations can get that same experience that we do. But artifact conservation is really complicated. Conservators do their best to stabilize artifacts by filling in the spaces that have rotted away with something else. One way is through silicone polymerization, which basically means they fill the artifact with a silicone plastic. This is really effective, but it's also irreversible. And unfortunately, sometimes even conserved artifacts start developing new problems. One problem is sometimes submerged wood can start generating sulfuric acid, which is the same acid found in most car batteries. So if you felt like that sounded bad, it is. The iron left over from nails in the wood reacts with sulfur, which comes from the water, to generate the acid, and it only gets worse over time. This can happen even with really unique artifacts, like this 17th century gun carriage from the La Belle wreck. La Belle was a French ship intended to colonize the mouth of the Mississippi. Instead, it sank off the coast of Texas. So it's really important for understanding and connecting with the early colonial history of Texas. But even though the gun carriage and other artifacts were treated with the silicone plastic, they've still been deteriorating for 15 years, and it's only getting worse. So what do you do when you have an irreversible conservation method and an artifact that's still degrading? Because usually you would undo the conservation so that you could get to the iron and remove it so it can't react with the sulfur anymore. In this case, you can't do that. So that's where my research comes in. I used samples and tried a couple of different common iron-removing chemicals to see if any of them could do the job, and found that actually one of them was able to remove the iron from around the plastic without damaging that plastic or the wood, which means that can be used to save the gun carriage and other artifacts like it. Now that we have a way to prevent that sulfuric acid from forming, hopefully we can save this and many other artifacts so that future generations of children and adults can get that same physical connection that's so important for getting history. Thank you.
We got a lot more competition to go, including the final competitor in the master's division, Julia Tapilatu from the Department of Ecology and Conservation Biology. Julia.
Imagine you're swimming in the big blue ocean, then somebody shouted, "Shark!" What's the first thought? Well, for most people, they will be scared. But do you know what's scarier than this? It's the fact that humans kill over 100 million sharks each year, yet they only attack less than 10 people annually. And despite of their scary look, sharks play an important role for our ocean. For instance, is the zebra shark, or Stegostoma tigrinum. They play a role as mesopredator in maintaining a balanced food chain. But due to their slow maturity and low production rate, they are vulnerable to extinction. A lot of human practices, such as overfishing, habitat degradation, continue to decline their population. And today, they have been listed as endangered species. If this condition continue to happen, we are putting our ocean to risk, where the absence of shark can lead to the collapse of marine ecosystem, impacting fisheries, economies, and communities. Immediate action is needed to prevent this from happening. In 2020, I was fortunate to join a zebra shark conservation program called the STAR, Stegostoma tigrinum Augmentation and Recovery Project. This program aims to reestablish a healthy population of zebra shark by reintroducing captive bred zebra shark to their natural population in my hometown, Raja Ampat, Indonesia. As the first project, research is essential to support reaching their goal. Therefore, my study came to place providing data about the growth rate and factors influencing it. It is important to know about this information so we can later understand how the population adapts to the changes of environment. And as organisms tend to grow differently in different environments, I hypothesized that different water facility systems can create a unique environment impacting their growth. And to test this, I collected data from different hatcheries and aquariums, then do a statistical analysis. The result shows that facilities with the natural source water system are more varied in their condition due to the changes of weather and season and human activities in the area. Also, the sharks that grow in the natural source water system are 3.7 times more faster growth in average compared to the recycled water system, where the dissolved oxygen played a key fundamental role here. So we can see that environment really play a key role impacting the growth of zebra shark, right? So hopefully from this finding, we can pave a better recommendation for the facility improvement, not only to safeguarding our zebra shark population, but also saving our ocean and future. Thank you.
Kick off our doctoral division, we have Siun Lee in the Department of Agricultural Economics. Siun.
Imagine you have just checked your investment portfolio and it's down, a loss. What do? Sell? Hold? Or maybe even invest more, hoping to recover. Most of us, when we're faced with losses, have an instinct to chase them, hoping to get it all back. This tendency is called myopic loss aversion, a short-sighted bias where we focus on immediate losses rather than long-term gains. In today's digital world, with constant updates on stock prices, it's easy to see how investors might fall prey to these biases. Myopic loss aversion combines two concepts in behavioral economics: a loss aversion, where losses feel more impactful than gains, and mental accounting, where we mentally separate our financial outcomes. But what happens when groups, rather than individuals, make these decisions? Do they also fall into the same trap? That's the question my research set out to explore. I tested whether groups exhibit myopic loss aversion using an investment game. In this game, participants were divided into two treatment groups. One that was forced to realize their losses after several rounds, and another that continued investing without directly facing their losses. Each participant played a game as an individual first, making an investment decisions over several rounds with the real chance to win or lose money. Then they were grouped into pairs and played again, this time making decisions collectively. So, what did we find? We found that both groups and individuals exhibit myopic loss aversion. However, the effect was stronger in groups. Groups are more likely to chase after their losses than individuals. This suggests that in a group setting, the urge to recover their losses is amplified. Group members might reinforce each other's biases, leading to a greater collective short-sightedness. This has a significant implications in financial decision-making. When people make decisions together, they don't necessarily become more rational. In fact, they may be even more biased. Understanding this can help design better investment strategies and encourage focus on long-term goals. So, next time you're tempted to check your stock prices 100 times today, ask yourself, are you investing for today's losses or tomorrow's gain? Thank you.
So we are pleased to move on to competitor number 5 this evening, who is Saptarshi Biswas from the Department of Biomedical Engineering. Saptarshi.
Imagine a soldier taking a bullet for his country and is unable to get proper patient care, which can cost their life or damage an organ for a lifetime. Surprisingly, till now, blood loss causes more than 80% of battlefield casualties and 40% of death in civilian setting. Surprisingly, the problem with the blood loss is that the treatment gets more challenging when the injury occurs in the internal body organ, lacking a surface to put pressure on. These injuries are not accessible to conventional pressure dressing, eventually increasing the mortality. Therefore, there is a pressing need to develop an alternative strategy to combat hemorrhage in the internal body organ. Now, what if I tell you that being a biomedical engineer, we have the power to create something that can save lives? In the Gaharwar lab, I am developing a combinatorial strategy using a foam and a smart biomaterial-based matrix to combat hemorrhage. Now the question is, what advantages does this system offer? First of all, due to the rapid expansion of this foam, the composite will fill in the wound cavity, providing a surface to apply pressure dressing. On top of that, due to the presence of this biomaterial-based matrix, it will help to concentrate blood clotting factors as well as activate the blood cells at the same time. I hypothesized that due to the synergistic effects of both foam and the biomaterial-based matrix to fabricate the composite, it will clot blood in a much faster way, and the results are promising. I observed an incredible 5 times expansion of the composite within 3 minutes while activating different blood cells. And this composite degrades much faster, as well as we did not see any detrimental effect of this composite when in contact with human skin cell. Upon delivery of this composite in animal model, we observed an incredible 80% reduction of the clotting time, and the blood loss also reduced to 50%. That is fascinating, right? Making it an excellent hemostat. Now, bacterial infection is very closely associated with hemorrhage. Therefore, while developing a futuristic hemostat, maybe clotting won't be enough. That got me thinking and is pushing me to driving my research to evaluate the wound healing efficacy of this formulation. Now imagine an all-in-one product with excellent hemostatic and wound healing ability, ready to help patients in both battlefield and civilian setting, and making it an alternative therapeutic strategy for better clinical patient outcome. Thank you.
We are ready for competitor number 6, and to get the prize wheel out of the way, our competitor number 6 from the Department of Biomedical Engineering, Zahra Ghiasi. Zara.
Vaccines. They are one of the most important health interventions we have ever developed. In fact, they are often considered as essential for public health as clean water. Recently, mRNA vaccines, like the ones developed for COVID-19, have revolutionized the way we fight pandemics. But how do mRNA vaccines actually work? We know that in conventional vaccines, we inject an inactive or weakened form of the virus into the body to trigger an immune response. In mRNA vaccines, on the other hand, we inject the mRNA, the genetic code of the virus, into the body. Our body then uses this genetic code to produce a small, harmless piece of the virus. This teaches our immune system to recognize and fight the real virus if we are ever exposed to it. mRNA vaccines are easier to produce, they work faster, and they are safer than conventional vaccines. However, there is a major challenge. mRNA is extremely fragile. It breaks down quickly and has to be stored at ultra-cold temperature to be stable. For example, the Pfizer COVID-19 vaccine needs to be stored at negative 112 degrees Fahrenheit because it's prone to degradation. This requires a special and expensive freezers and a carefully maintained cold chain. Well, it creates serious challenges in how we store and how we distribute vaccines to different locations, particularly to low resource areas where this super cold storage simply isn't available. Here is where my research comes in. I'm focused on making mRNA vaccines more stable. By using experimental and computational tools, I modify the mRNA molecule itself. Basically, I optimize the shape and structure of this molecule to make it more resistant to breaking down. This helps the mRNA last longer, allowing the vaccines to be stored at higher and more practical temperatures. Additionally, these modifications also make the vaccine more effective by boosting the immune response with just a single dose. Ultimately, my work could lead to vaccines that are more effective, more durable, and easier to distribute around the world, helping us stay one step ahead in the fight against future pandemics, no matter where in the world they occur. Thank you.
All right, our next competitor from the Department of Mechanical Engineering is Aditya Yalamanchili. Aditya.
I'm assuming most of us here drove our cars to campus today, but did you know that the sheet metal in your car, from the body panels that protect you to the copper and electric vehicle batteries, is a significant contributor to climate change? That's right, the way we make metal sheets, whether steel, copper, or aluminum, hasn't changed much in over a century. We heat large slabs of metal, roll them, cool them, heat them again, roll them again, cool them again, and repeat this cycle over and over until the desired thickness is reached. Imagine, if you will, kneading dough, but if it were metal. The process is energy intensive, expensive, and contributes significantly to global emissions. In fact, steel production alone accounts for about 7% of the global carbon dioxide emissions, which is more than the entire aviation industry combined. But here's the exciting part. There is a better way. My research is exploring an energy-efficient alternative, a single-step process for producing metal sheets by peeling a thin layer directly from the surface of a solid metal stock using a knife edge. Think of it like peeling an orange, but instead of fruit peel, you're creating a precision, high-quality metal sheet. The picture here on the slide shows our peeling process in action, a sharp edge smoothly slicing a sheet of metal from a solid block. So here's why it matters. This method drastically cuts down energy and carbon dioxide emissions. The global, I'm sorry about that. So we need to control the thickness of that peeled sheet. Think of peeling the orange again. If you pull the peel too erratically, it's going to come off thick and uneven. But with the right amount of steady force, you get a thin, continuous strip. My research focuses on developing the manufacturing systems and the control strategies that ensure that the thickness, speed, and tension of the peeled sheet remain uniform as it is peeled and transformed into a high-quality coil. Using this technology, we have been able to produce metal sheets thinner than 100 micron. That's about the thickness of a sheet of paper, perfect for new generation applications such as battery foils, and with equal or better quality compared to traditional methods. This breakthrough can potentially reduce over 1 billion tons of carbon dioxide emissions by 2050, which is equivalent to removing hundreds of millions of cars from the road. Metals power our lives, but their production shouldn't cost us the earth. My research offers a sustainable solution ready to meet the world's growing demand for metals toward a future where technology and sustainability go hand in hand. Thank you.
After that, it is time for competitor number 8, who is Poojashree Chettiar from the Department of Psychiatry and Behavioral Science in the College of Medicine. Poojashree.
How many of you had a heartbreak? Yes, we have all been there, listening to the same sad song, scrolling through old photos, unable to let go. A feeling of being stuck. That's your brain struggling with behavioral flexibility, the ability to adapt and change. Now, what if that stuck feeling wasn't just about a heartbreak, but how your brain handles every decision? For 1 in 8 people with neuropsychiatric conditions like addiction, OCD, or autism, this is their daily reality. Sadly, 60% of them don't receive any proper treatment. With this motivation, I set out to identify molecular mechanism in the brain driving this repetitive behavior. By recording, imagine your brain as an orchestra. Instead of music, it produces memories, thoughts, and movements, with proteins acting as an instrument. When even one of these proteins malfunction, it's like a whole orchestra falling out of sync. But I have identified 2 key proteins, GLUT1 and PKC, which works together to maintain this orchestra in a harmony. GLUT1 forms a bridge between the neurons, ensuring they communicate effectively, while PKC acts like a therapist deciding whether this communication should be strengthened or weakened. By recording neuronal activity, I have found that in most of the brain, PKC activation strengthens communication. However, in the striatum, where GLUT1 is present, it weakens them, leading to synaptic depression, just like a therapist asking you to let go. Further, to explore this, I used molecular technique to delete GLUT1 from specific brain regions. Without GLUT1 in the striatum, a region crucial for movement and learning, animals struggled to adapt in a task where I initially trained them to find a platform hidden in water and then changed its location. Now, they had to relearn this new path. But they kept circling aimlessly, just like a relationship stuck in a loop. This confirmed the malfunctioning protein
as well as the location of disruption. But I didn't stop there. By combining insights from existing studies with my own findings, I have identified a site within GLUT1 that might regulate this PKC's function. To test this, I developed a tool that mimics this site of GLUT1. Delivering it directly into the striatum of the mice improved their learning and adaptability by almost 80% compared to the untreated mice within just 6 hours of treatment in the same experiment where mice had to relearn a new path. We all know the feeling of being stuck. My research provides the brain the tool it needs to break free, giving hope to millions of people fighting neuropsychiatric conditions. Thank you.
And I will reappear after our 9th competitor from the Department of Soil and Crop Sciences, Sarah Chu. Sarah.
If you dropped 1.2 million pennies on the ground, would you pick them up? I would try because money is money, but it would be annoying. Now, what if those pennies were actually weed seeds, smaller than the size of a sesame seed? Would you pick them up then? Or would you figure out a way to prevent them from ending up on the ground to begin with? This 1.2 million seeded nightmare is real in the form of Palmer amaranth. A weed that originated in the desert has adapted to multiple different climates and areas in the US. It has also developed herbicide resistance, sort of like how our bacteria has become resistant to antibiotics. This means farmers, specifically cotton farmers, need a new way to manage this weed. And I suggest we do that by targeting the seeds themselves. I'm looking at how a cotton harvester may capture the weed seeds, let the weed seeds remain on the plant itself, or they may end up shattering to the ground during that process of cotton harvesting. I have found that over half of the 1.2 million seeds are actually captured with the cotton harvester. That means our cotton harvesters are not weed seed spreaders. They're in fact weed seed catchers. This is great because that means the farmer is removing those seeds from the field. But if we look at what happens next to this cotton weed seed mixture, it actually goes to the cotton gin, which is where our cotton gets cleaned and all that trash weed seed mixture actually ends up going into a pile in the back. That pile gets spread back out onto farmers' fields. So that means they're reintroducing their weed seeds once they remove them. So I propose that we sort of target those weed seeds while they're present in that pile, and I did just that by using an impact mill, a type of machine that crushes and pulverizes weed seeds and all that plant material. I found that it killed 99% of these weed seeds, which is great. With the implementation of this technology, farmers no longer have to worry about spreading 1.2 million seeds in their fields. Instead, they know their cotton harvesters catch and later on can be used to kill these weed seeds, in fact, instead of catching and releasing them. And that really means that they can stop it in their tracks and stop further spread of this weed throughout the US. As we know, no seeds means no weeds for years to come, and it will save them in the long run. Thank you.
Our next finalist is Tatlock Lauten from the Department of Medical Physiology. Tatlock.
I want you to try to remember a time when you were truly terrified. What did you feel in those moments? Was your heart racing? Did your breathing become quick and rapid? Did you get that familiar pit in the bottom of your stomach? For many of you, these feelings are just memories, and they do nothing to you physically right now. However, for 24 million people living in the US, they relive these feelings on a regular basis, which, as you can imagine, significantly impacts their ability to live their normal everyday life. This phenomenon is called post-traumatic stress disorder, or PTSD for short. PTSD occurs when the body's alarm system otherwise known as your fight or flight response system, continues indefinitely after a traumatic or a fearful event. This long-term activation leads to the behavioral aspects that we know about PTSD, including increased anxiety and flashbacks. Now, I really want you to focus on what I tell you next, as it's really important. PTSD does not just affect the brain. In fact, the immune system, normally responsible for fighting off infection and disease, is kicked into overdrive during PTSD and starts attacking its own body, leading to development of autoimmune diseases such as rheumatoid arthritis, psoriasis, and multiple sclerosis. This is where my research focuses, how the brain influences the immune system. In order to study this, I developed a new mouse model that allowed me to systemically target various drug and genetic aspects. In addition, it also allowed me to compare the differences between men and women who undergo PTSD. In my research, I found three main findings. One, the brain sending a signal to the immune system, that signal is called noradrenaline. Two, by repurposing an FDA-approved drug for high blood pressure, I can limit the amount of noradrenaline that is seen by the immune system. And three, by limiting the amount of noradrenaline that gets to the immune system, I can greatly reduce the damage done to the body. This is important as presently therapies for PTSD are limited, and none of them target the immune system. This is why it is my goal that my research will be used as a new therapeutic avenue for patients with PTSD. By treating both the mind and the body, we can give these patients back the sense of normalcy and healthy life that they used to have by giving them this drug tomorrow, ready off the shelf. Thank you.
From the Department of Soil and Crop Sciences, Dallas Williams. Dallas.
August 25th, 2017. For most Texans, this was an ordinary day, but for some along the Gulf Coast, it was life-changing. This is the day Hurricane Harvey made landfall in Houston, starting what would become one of the most catastrophic flood events in U.S. history, with max floodwater depths reaching 12 feet and resulting in more than 100 deaths. 1.5 billion gallons of water, an amount of water that could fill Kyle Field completely to the top, not just once, but 22 times. This is the amount of floodwater New York City is able to reduce each year through its network of green infrastructure, which includes thousands of rain gardens that capture stormwater runoff which is generated when the ground can no longer absorb rainfall and it accumulates on the surface, causing flooding. Now, some of you may or may not know, the Texas state soil is a heavy clay that resists drainage and is very prone to flooding. Therefore, when making a rain garden in Texas, this soil is removed and replaced with sand. But did you know the sand used for these purposes is mined? And according to recent research, the global demand for sand will far outweigh the supply in coming decades. This inspired me to find a way to make rain garden design in Texas more sustainable. Last summer, I conducted an experiment in a greenhouse where I tested the performance of 4 different rain garden designs. 3 of those designs contained a waste product, ordinarily destined for the landfill, either crushed glass, crushed oyster shell, or expanded shale, each mixed with clay soil. The 4th design contained sand. I created mini rain gardens in PVC pipes that were 3 feet long and 6 inches wide. Then I performed 6 separate tests where I applied approximately 1 gallon of water to each of the designs and recorded how long it took the water to disappear into the surface and drain from the bottom. My results showed there were no significant differences between any of the designs, indicating that mixing either glass, oyster shell, or shale with clay soil can reduce similar volumes of floodwater as using sand. Flood events are becoming more frequent and more extreme, as we have seen with the recent devastation along the East Coast. Making our cities more resilient to flood risk is more important than ever before. Luckily, solutions don't need to be complex or expensive, they just need to work.